Hi everyone, this is Diana, and today I'm going to talk about receptors and molecular signaling in neuroscience. So a receptor is a protein that a signal molecule can bind to in order to prompt some type of cellular or physiological response. To break down this definition, we consider the diagram on the right. The ligand, which is a fancy term for the signaling molecule, is binding to the receptor in the cell membrane, which is the barrier of the cell. This receptor is activated and starts to issue instructions within the cell until a response is finally issued. Receptors are commonly found in the cell membrane because it is difficult for many molecules to cross the membrane into the cell by themselves. The insides of the cell membranes are hydrophobic, which means that they repel charged molecules. However, molecules that can cross the cell membrane easily, like steroids and lipids, can have receptors inside the cell because they just travel across the cell membrane and go straight to the receptors. So we mentioned that ligands bind to receptors, but what types of ligands? In neuroscience, most of the time we're going to discuss neurotransmitters, but we also have neuromodulators, which are similar to neurotransmitters, but they bind to a different location on the receptor protein. Many proteins have an active site, which you can see here, where the main ligand binds to, but some of them also have an allosteric site where other molecules can bind to in order to regulate the protein's activity. So here we have an allosteric activator, which is going to help the protein perform its function, but we can also have allosteric inhibitors, which will basically turn the protein off. So neuromodulators focus on binding to the allosteric sites of their receptors, whereas neurotransmitters bind to the active site. Another category of molecules that we have here are prostaglandins, which are small lipids that are made when body tissue is damaged or infected, and they can increase inflammation, pain, or fever while they're healing the damaged tissue. These molecules also need to bind to receptors in order to carry out the actions that we just mentioned. And we can also think of hormones. Hormones bind to receptors in order to initiate their responses. For example, adrenaline binds to your receptors in order to increase your heart rate, blood pressure, among other things. So we gave some relatively vague descriptions of ligands and receptors, so we're going to talk about a specific type of receptor, which is the ionotropic receptor. It's also called a ligand-gated ion channel. With this type of receptor, a part of the protein acts as a gate that controls the entry and exiting of ions. And normally the gate is closed, but when the ligand binds to the receptor, the gate opens and ions can now flow into the cell to prompt a response. The second type of receptors are metabotropic or G-coupled protein receptors. These are bound to a G protein that is normally inactive. When the ligand binds to the receptor, the G protein activates and detaches from the receptor to initiate more cellular responses. For example, here we have the G protein breaks off and activates another enzyme. And if we see this animation, we'll get another example. So the ligand has just binded to the G-coupled protein receptor, and now the G protein is starting to break off. We'll see soon what happens with it. Okay, so a part of the G protein has now binded to this ionotropic receptor and activated this receptor, and the other part can perform other functions. Here it's activating another protein which will initiate a signaling cascade, which is basically like a domino effect. This enzyme will activate another enzyme and so on and so forth. So we can see that metabotropic receptors are a bit more complex than the ionotropic receptors we just looked at. They can initiate multiple responses, and that's why they're considered the slower type of receptors. Now we'll briefly discuss the signal transduction pathway, which is a fancy name for how a cell will interpret a signal detected by the receptor and convert the signal into an actual response. Finally, the ligand has binded to the receptor, and we can just consider that this is a G-coupled protein receptor because it has this protein, and now that the protein has been activated, it's starting to activate even more enzymes, and each of those is activating another enzyme. So we get what's called the transduction part of the pathway, where multiple enzymes are being cued and multiple processes are occurring. And so here, the end result is that a gene is expressed, but this is not always the end result with signal transduction pathways. As we saw with the metabotropic receptors in the previous slide, there can be many possibilities for the end response. And now, to put some of the concepts we learned to practice, let's look at the neurotransmitter acetylcholine and its receptors. Acetylcholine is an excitatory neurotransmitter involved in muscle contraction, memory, and cognition. Acetylcholine is commonly deficient in patients with Alzheimer's disease, which makes sense considering that we just mentioned it plays a role in memory. Acetylcholine has two main classifications of receptors: nicotinic, which are ionotropic, and another classification that are metabotropic that we won't discuss in this presentation. And all of these receptors are called cholinergic receptors, which just means that they're receiving acetylcholine. So we mentioned that acetylcholine is involved in muscle contraction. Acetylcholine is found at the neuromuscular junction, which is the connection between a motor neuron and muscle fiber. When the muscle needs to contract, acetylcholine is released by the motor neuron into the synapse, and it will bind to the nicotinic receptors. These ionotropic receptors will open up their gates so that sodium can flow in and potassium can flow out. And because more sodium flows in than potassium flowing out, the muscle cell will be depolarized and will be stimulated to contract. Now let's look at another neurotransmitter, glutamate. Glutamate is the most common excitatory neurotransmitter, and it's involved in learning and memory. It's also considered the main excitatory neurotransmitter in the central nervous system, or CNS, which is a very important fact to keep in mind. Glutamate also has many types of receptors, including three main types of ionotropic receptors: NMDA, AMPA, and kainate receptors. Glutamate also has metabotropic receptors, but we won't talk about those in this video either. So now let's take a closer look at NMDA and AMPA receptors. Both of these are ionotropic, but NMDA produces slower synaptic responses, whereas AMPA receptors are fast and brief. I included a few more differences between the receptors, but they're not super important. NMDA receptors allow potassium, calcium, and sodium ions to flow in, while AMPA receptors only focus on generating a potassium and sodium influx. Both receptors are also involved in synaptic plasticity, which is the process by which synapses strengthen and weaken. And this description is a little bit confusing, so let's take a more specific scenario. Let's say you're learning how to do algebra. Certain networks of neurons in your brain are going to focus on learning and performing this task. And the more that you perform it, the more glutamate and excitatory neurotransmitters will strengthen these networks and the synapses that connect all of these neurons, which is how you're able to learn. On the other hand, when you've stopped doing algebra or a task that requires practice for a few years, the synapses might start to weaken because they're not being used. And this whole process of synaptic strengthening and weakening is due in big part to these NMDA and AMPA receptors for glutamate. This is also why Alzheimer's has been connected to a dysfunction of the NMDA and AMPA receptors. For our last example, let's look at GABA, which is an inhibitory neurotransmitter. When GABA binds to its ionotropic receptors, the gates will open to allow chloride to flow into the cell. If you remember, or if you can see here, chloride ions are negatively charged. So the more that they flow into the cell, the more negativity will accumulate on that side of the membrane, on the inner side of the membrane, and the less likely that an action potential is to fire, which is why GABA is referred to as inhibitory. Alright, that's it for this video. Let me know if you have any questions or if you want to know more about this topic. I've also attached some more videos in the slides that involve reception and molecular signaling, so feel free to watch those and to ask all the questions that you have.